This is Sam Salami of Device Talks. Welcome back to our materials and manufacturing webinar series brought to you by Device Talks. We've got a very hot company here to talk about a cool new approach to structural heart valve manufacture and materials. You can see the title there, Reinventing Heart Valves, Driving Innovation with Life Polymer and Robotic Manufacturing. The company is Foldax. We've had them recently on our podcast and also at Device Talks West. Very happy to have them here today presenting live to you. Thank you, of course, to our sponsors, Bokers, Jabil, PSN Labs, Mitsubishi Chemical Group, Sekisui, and Forge Medical for making this entire series possible. Really grateful for their support. Thank you for joining us live today. If you are not joining us live today and you're watching us on demand, enjoy this presentation. Folks who are watching live, you'll be able to ask questions through our Ask Question widget. That's the little Q&A box there. Submit that. We'll likely collect them all for the end. We've got a really full presentation, lots of great data and imagery for you to enjoy. So we'll let our two experts kind of roll through their presentation and then get your questions at the end. We also have the slides available to you in the resource box, so you can check that out there. And sorry, just looking at got a text chat box and trying to decipher it. So enjoy this presentation. Once again, live attendees, we love the emojis. Let us know we're doing a great job by giving us the thumbs up or a smile. Whatever your heart says, share it with us. We'd love to know you're out there and enjoying the conversation. And real quick, I'm going to run through this. There's the heart. Thank you so much. If you're we've got a lot going on, so I'm going to stop my usual tap dance and just get right into it. But if you're having a problem, push refresh. That normally does the job. You can help yourself by closing all the other apps and browsers to make sure your bandwidth is you're giving as much of your bandwidth as possible to this presentation, plus giving as much as your attention as well. I think it's a real important one you should be focusing on. And if you still need help, you can try the little 3-line burger icon there, but just ask our great producer, Amira Weininger, for help in the Q&A box. She'll get right to you directly and make sure you're able to hear everything and see everything. And once again, this will be available on demand. So if you are watching this and realize that someone in your circles would enjoy this conversation, let them know that they can go to devicetalks.com to register. While you're there, check out our great podcast. You can also register subscribe to our podcasts on any major podcast player. We've got a lot coming at you, including our recently launched Women in MedTech podcast, hosted by our managing editor, Kayleen Brown. So once again, thank you to our sponsors, Bokers, Jabil, Sekisui, Mitsubishi Chemical Group, Forge Medical, and PSN Labs. And without any more jibber jabber from me, I'm actually going to introduce our two panelists. We were able to bring in an additional expert for you to enjoy. So very happy to have Ryan Stanfield, who's Vice President of Engineering and Operations at Foldax, and Jim Runt, he's Professor Emeritus at the Department of Material Science and Engineering at Penn State University. He's also on Foldax's Scientific Advisory Board. So I'm going to, I think, hand things off to Ryan to kick us off. Then they'll bounce around, and I'll be back later on when the questions start. So Ryan, thanks for giving this presentation today and for sharing Foldax's story.
Yeah, of course. Thank you very much, Tom. Thanks for this opportunity. Really appreciate it. And thanks to everybody at Device Talks for organizing it and having us on. With that said, I will just start out and talk about Foldax and what we've done and what we're doing and our vision, for the future with automation. And, that's robotic manufacturing of polymeric heart valves. So, when starting this and throughout all of this process, we've been asking ourselves, some of these questions of, what if we had a material that was a catalyst for change, and specifically a change in the structural heart industry? This could potentially break barriers, that are held by current platforms with biologic tissue or mechanical valves and the materials that they use. It could also, we could also use design tools specifically for polymer and for other materials to optimize the valve. And the big one that's of a lot of interest to me and many others is automate manufacturing and take out a lot of human variability in the assembly and manufacturing process, and ultimately deliver a big change to patients worldwide. There are a lot of patients, and we've seen that happen so far in our clinical trials. And so we'll talk a little bit more about that. So we're going to go through and talk through, heart valve prostheses, first of all, and then I'll hand it off, and Dr. Runt can talk about polymer material, and then I'll come back and talk about, our design of valve with computational tools and with polymer, and how we do robotic manufacturing and some of the work that we've done clinically to date. So that being said, I'll get right into it and talk about just heart valves and some of the barriers that we have and what we're trying to do with current materials. So there's this balance back and forth of, in surgical valves, but also there are transcatheter valves. But in surgical valves, there are tissue and mechanical valves. And so tissue valves have a lot of great things, and so do mechanical valves. With tissue valves, they have a really good quality of life, and other options with surgical recovery. And with mechanical valves, there's a lot of great things associated with those. They have a really high durability, long life. However, patients need to be on anticoagulation basically for their entire life. There's a higher manufacturing cost to those. Just due to the design, reintervention or reoperation, putting a valve inside of a valve, that's not an option. That is an option with tissue valves. But with tissue valves, there's also a high cost to manufacturing. There's a lot of manual procedures and processes there. There's harvesting of tissue and, matching that, making sure they get good leaflets that match one another. And so, we have this vision and idea of, can we take a novel material and apply it and overcome some of these challenges that are in both of these different types of heart valves, both tissue and both mechanical. And so with that being said, Dr. Runt's going to come in and talk about polymer material and both a background of polymers and what's happened and what we're doing going forward. So with that, over to you, Jim.
Thanks, Ryan. Very happy to be here. One of the things we'll start off with some background for everyone who may not be familiar with the demands and the history of polymer heart valves. The life polymer that we're referring to is a very unique material that was designed with the following 7 items in mind prior to the synthesis and creation of the material, as opposed to pulling something off the shelf and hoping that it works, right? So what do we want? Think about a heart valve. We want good physical properties such as resistance to tear propagation, certain dynamic modulus. UTS there means ultimate tensile strength. You want it to be stable in the body, of course. You want it to be resistant to mechanical deformation, that's creep. You don't want it to creep. It's got to be blood compatible. And it should also be non-calcifying, which causes a lot of problems, serious problems in heart valves, if you get it. So non-toxic and also no leachables. This is a tall order, right? And also, last and not least, manufacturing stability and manageable supply chain. Okay. So let's go back just a little bit and think about the history of synthetic polymers in biomedical applications in general. And this started back 75 years ago with people trying silicon rubber, PDMS, as a biomaterial in the body, and the body tolerates PDMS very well, but it has terrible mechanical properties. It has very low tear resistance and so on. And through the 50s, 60s, 70s, people were taking other materials off the shelf and trying to use them in certain biomedical applications. Some worked, some didn't. And then as you go from 6 o'clock on, there are variations of those materials modified with carbon nanotubes and cross-linking and so on. And you get up to about 11 o'clock. It takes us to, 2015-ish. You see Life Polymer there, SIPUU. That's an acronym for it. It's a little bit more complicated, but it's a polyurethane. And it was designed with those 7 criteria in mind on the left-hand side. So that's the unique aspect. We didn't just take something and try to use it. We designed, the people who did the chemistry designed all of these characteristics into the polymer, ahead of time with those in mind. So what's a polyurethane for those who are unfamiliar? It's what's called a multi-block copolymer. That doesn't matter for our purposes here, but it's composed of 2 different segments. The red segments on the top, the cartoon on the top, and the blue segments. The red segments are so-called hard segments. They have high glass transition temperature, they're stiff. And then so-called soft segments, which are flexible. They have low glass transition temperature. And they're in the same chain. So both the chains are composed of hard segments and soft segments. And when you manufacture this and you create a solid material from it, what happens is the hard segments segregate away from the soft ones. And so you end up with a structure, it's called microphase separation, but it's actually nanoscale phase separation. If you see the cartoon on the lower left, the red dots there, and they're certainly not uniform in reality, they're on the order of nanometers. And the blue continuous phase is flexible. So you end up with no chemical crosslinks, with thermoplastic hard domain crosslinks. So in principle, you can melt the material, certain materials, and reformulate. The slide on the right is actually not very clear. It's an older atomic force micrograph of a typical polyurethane, basically distinguishing the brown background from the yellow dots there. The yellow is the hard segments, and background is the soft segments. There's some better images I could have used there, but you get the idea. So we're getting segregation of the blue away from the red. So what are we worried about? What we're most worried about is biodegradation, because what happens when you take, whether it's a heart valve or any other implant, what will happen immediately is you have white cells that'll adhere and fuse into the foreign body being the biomaterial. Those cells will excrete oxygen radicals, enzymes, which could cause hydrolysis and oxidation and failure, sometimes in a relatively short period of time. So polyurethanes do have tunable mechanical properties because you can change the amount of red and blue, and they generally have good tolerance in vivo. Let's see if I can. I got this is next. Okay. But the situation, and you'll see this a little bit later in the presentation. A conventional low modulus or flexible polyurethanes, they tend to degrade on long-term implant. And the soft segment, the blue segments, are the most susceptible sites for degradation. I keep hitting my own buttons. There we go. And this is just one example. They're not very clear, but it's a cell-mediated degradation of a soft polyurethane. Left-hand side, it's really hard to see. What you're seeing is a scanning electron micrograph of cells on the surface of a polyurethane biomaterial. And when you take those cells off, what's underneath is a whole bunch of pits and cracks and damage, which are sites for crack propagation and ultimately failure. Right. So going back to, I guess, certainly back 20 years ago, people were searching for ways to use different kinds of soft segments that are less susceptible to damage, oxidation, and hydrolysis. A lot of these soft segments at the time were based on polyether chemistry. In other words, it has a couple of carbons and an oxygen, and people tried to, and very successfully, to some sense, in some sense, to increase the hydrocarbon concentration in the soft segments and create more stability. And that works up to a certain point, but not completely. And this is out of sequence, but you can see the damage that's actually done with a soft polyurethane at a somewhat higher magnification. Okay, so that kind of sets the stage. We've got a problem. We have materials that are flexible. They are suitable for heart valves in principle. In practice, they're not stable enough. So this slide gives you a bit of the background for what gave us the inspiration for Life Polymer. I'm using us in a very generic way. In the 1990s, well, let me back up a second. Prior to that, people would thought about, can I take silicon rubber, siloxane, and connect it to polyurethane segments and give me a mechanically robust but biocompatible, biostable polymer? But they couldn't find a way to synthesize it. They could make a few thousand molecular weight, but didn't have any mechanical properties that were useful. And the breakthrough was in the mid-1990s at CSIRO, which is a research institution. This site was in Melbourne, Australia. And they found a way to incorporate, and again, see the cartoon on the right-hand side. It's not so important for our purposes. We got hard segments and soft segments, with the blocks for hard, meaning rigid, and the squiggly lines for soft. Found a way to couple soft segments, siloxane soft segments, with polyurethane segments. And the key, the magic dust, was to use a compatibilizer, a chemical compatibilizer that permits the stable connectivity of siloxane soft and urethane hard segments, right? So the siloxane chemistry is shown in the blue box. And the compatibilizer is a simple polyether, which has a pretty long hydrocarbon segment in there. So that ends up at certain compositions. First off, the flexibility is you can create various compositions, but at certain compositions, it has properties such that it's been used. It was first spec'd out, of course, and ultimately used for pacing leads, insulating pacing leads. It's been on the market for 20 years under the trade name originally of Elastion by Aortec, and now Optum by Abbott. Abbott has implanted 7 million pacing leads now, with 0.1% breaches over 20 years. And not all those breaches are polymer related. In fact, it's not clear that any of them are polymer related. So this material is really stable in a pretty aggressive biological environment. But this material is too stiff for heart valves. So what did we take away? We learned how we can connect siloxane segments with urethane segments using a compatibilizer, but we need to modify the chemistry further. So this is how Life Polymer came about. It's uniquely formulated for the properties necessary for heart valves from the ground up, not taking something off the shelf, but from the ground up. It has inherent properties that are well suited for heart valve applications. So what's the next step for those who are interested in the chemistry at all? Life Polymer was developed specifically. It used that basic siloxane urethane structure And you can see on the top right the cartoon up there with the compatibilizer, the siloxane and the hard segments. And then it incorporates patented other chemistry that has other chemical groups in there, like ureas and a second chain extender. Again, that's not so important, but you end up with desirable properties for heart valves while maintaining a very high concentration of the siloxane units for biostability. So you're getting good mechanical properties, you're tuning in the characteristics that you want, and you're keeping the biostability associated with the siloxane units. So that's life polymer, right? Certain chemistries, a certain fraction of these components. It's got improved flexibility over Optum. It's got terrific resistance to tear propagation and no creep. Right? So no deformation under load. You don't want your material, your leaflet to change shape over time. So it changes the hemodynamics. So it was also, part of this was formulated to resist calcification. And there's a standard test that's done to assess calcification. It's sort of an accelerated test where either in rats or in rabbit models, sample is a dog bone or a strip of life polymer or any polymer is stretched to 150%. It's held there. And in this case, was implanted in rabbits. This was actually in Australia for 6 months. And we compared, you can't see it here, but we compared positive and negative controls to life polymer. The negative control was a material the positive control was the material that we knew would degrade in the rabbit in a very short period of time, in 6 months. The other material was a commercial material that's used by Medtronic as a pacing lead, very successfully in the pacing lead, but even that was damaged a little bit in the 6-month time period. This number, we see number 3 there. This is the pair of tweezers, and it's holding the sample after it's been taken out of the explanted from the rabbit, and it's pristine, right? And what we did was we did X-ray photoelectron spectroscopy. Again, what that is not important, but we gently cleaned the surfaces to remove the biological material, and then used this characterization technique, which basically is characterizing the elemental composition of the material at the surface. And you can't see the details there, but there's no calcium detected at the surface. Also from the clinic, right, after 3 years, this is an aortic valve, I believe. After 3 years of implant, we found, this the top right is not a very high magnification scanning electron micrograph of the surface. It's pristine, just like the others that we've seen. Then we use a slightly different characterization technique, energy dispersive X-ray diffraction, to again do elemental composition. And again, there's no calcium, right? So we're getting no calcification in the usual type of in vivo tests. And by the way, if we're interested in all this, very small font down the bottom left is a paper that we published back in 2021, and has all these details in there. So it doesn't calcify. Has good mechanical properties, and also you want it to be very resistant to platelet deposition. And so we used the common human primate model. This was some years ago. It's also reported in that 2021 paper. And what we did was to use this as a baboon shunt model to look for thrombus formation in life polymer, compare it to the baseline material. And so you can see a picture here, a photograph here of the splice where the material is placed in the baboon shunt model. And again, the details are shown down below in lower font. But the takeaway message is on the y-axis is the average platelet content on the material in billions. All right. Now it's just a linear scale. And then the time in minutes. So it's a relatively short duration experiment. The baseline material, which is known to be quite biocompatible and to be the kind of standard material that does not have much platelet deposition, is expanded polytetrafluoroethylene, so expanded Teflon. It's basically stretched Teflon, which is porous. The error bars are big, that's the red, but you can see that over a certain period of time, there's about 1 billion platelets on the surface. Same experiment, same time period, life polymer, much lower platelet composition. So it's very resistant to platelet deposition. Okay, I believe that's it for me. And I'll be happy to take questions later on. I think we're going to run through this. So Ryan, yeah, we're up to you now. So
great. Thank you, Dr. Run. I think that was very important for everybody to get a background on polymers and life polymer, because I'm going to go in and say, okay, now that we have a polymer that's designed and is customized for this application, what can we do with it now? And so with that said, we can take the life polymer and design leaflets, but also part of this is, we have a polymeric frame in our surgical valve that's a polyether ether ketone, PEEK. And you can see we use, finite element analysis to look at that, look at stresses and strains and optimize that and utilize the properties of PEEK to come up with a functionality in the frame that will also help the leaflets and the valve overall in its performance hydrodynamically, as well as for long-term durability. And so you can see that here kind of in the overlaid image that is at the bottom left, there is some deflection to these stent posts, if you will, for the valve at each commissure. And that aids in the valve function. And so as we apply the life polymer leaflets here, we know the properties of life polymer now. Dr. Runt described that very well. And so we can apply that in our in silico models and complete our full design here, look at optimizing stresses and strains throughout the leaflets as well as in the frame, and look at flow patterns and how we'll do that and become ready for validating these models. With that said, then we can, with that designed and modeled, we can take that and optimize each valve for each environment. So we make, right now, we make a series of aortic valves and a series of mitral valves. We make 6 sizes for aortics from 17 to 27 millimeter, and 5 sizes for mitral from 23 to 31. And so to my point is that the 25 millimeter aortic valve is not the same as a 25 millimeter mitral valve. The frame, the leaflet thickness have all been optimized for each specific environment. You have a different pressure gradient for each environment for aortic versus mitral. Also with this polymer, we can get it relatively thin, and reducing that thickness of the leaflet reduces the mass and therefore reduces the inertia. And so the valve leaflets can open and close faster than other valves. And so it makes the valve much more efficient and gives it much more longevity that again can last longer and hopefully require fewer surgeries for each patient. So that being said, we test this in vitro per ISO 5840, and we look at the effective orifice area and pressure gradient over time. We do this under an accelerated wear condition, and this is for one of our mitral valve sizes, the 27 millimeter, with a number of them on test. And this test, at accelerated, it takes about 2 years, and it simulates, almost 20 years, depending on how you do your math, 16, 17 years of real-time use. And so you can see here that the effective orifice area is stable over time, the pressure gradient stable over time. We also do this with our small aortic valve. So same thing here. This is, different size valve, but it's very stable over time in vitro testing under accelerated wear conditions. And so, when we look at it and we compare it side by side with, so this is compared to the Edwards tissue valve that you see on the right, and the Foldax Tria valve on the left. You'll see how kind of stable the leaflets are in the polymer valve as the valve is opening and becomes fully open in a round configuration, and then quickly closes. So if you run these side by side, there are differences, and you look at it with the timer, there are differences in how long each one is open under the same conditions. Similarly, we put it into an animal heart ex vivo, and so you can see this one's really cool. It's a video I really like. Right here's the aortic valve, and then the big one on the lower right is the mitral valve. So that's a shot from inside of the left ventricle. You can see both valves functioning on this ex vivo model. So I'll talk a little bit more now about the potential for these heart valves in patients and what we've done and what we're doing there. So we've got a couple of mantras here at Foldax. I would say 3 of them that are focused on reducing costs, both for cost of manufacturing, but ultimately cost for patients and payers. Injection molded valves, they cost a significant amount of time and capital to produce, even if you are using an injection molded polymer. And so we use a dip casting method. And so it allows for the leaflet geometry to be formed without a mold and no direct labor, training that with automation. And the computer algorithms that we use in computational design can remove a lot of testing and experiments that you do through design iteration. Ultimately, we do all of the testing that's required by ISO and by FDA. But in the design process, there's a lot that can be done computationally. And finally, our robotic manufacturing is an efficient, low-cost, scalable solution that can eliminate human interaction with the product and really get a good manufacturing supply chain as well as manufacturing process in hand. And so we synthesize our own polymer for this. We synthesize the life polymer that Dr. Runt talked about. So the valve itself is fully polymer. And you'll see that here. The frame, like I mentioned before, is polyether ether ketone. It's been made radio opaque, so it can be seen under visualization while it's in the body. The 3 polymer leaflets are dip cast as a single piece. So this isn't taking a sheet of polymer and cutting out a leaflet and then bonding it on or suturing it on. These leaflets are all dip cast into place on this frame. And then that same material, that life polymer material, encompasses or encapsulates the peak frame as well. And then a porous sewing ring that's also polymer is sewn on to the peak frame. So you got full polymer valve here. We take that and we just look at the benefits that this can have beyond tissue and beyond mechanical valves. With it compared to bioprosthetic valves, like Dr. Runt showed, it resists calcification. We've seen this in vitro and in vivo. There's no animal products that are used, right? No animal tissue, so it eliminates variability with the manufacturing process and with supply chain and using animal products. and it's has a really good structural integrity. We know, this is a relatively homogeneous material. We know the properties of it, so we can design it and optimize the design for the best stresses and strains as the valve functions. And, when we compare it to mechanical valves, there are no hinges, and so no clicking noise. When patients have mechanical valves, a lot of them can tell you they can hear the valve, and their loved ones can hear the valve opening and closing at night while they're sleeping. We use a tri-leaflet design here. So without any hinges, there's nothing in the, central orifice area. And so, something could go through it if needed, including, a valve replacement. There's a number of procedures out there called valve in valve, where they go through and they put a transcatheter valve into an existing valve that's been replaced. A little bit talk about here on our robotic manufacturing or automation that we do with this valve. So I'm going to show a really cool video. This video is also available on the Foldax website. You're welcome to see foldax.com. And it's going to run through this video kind of quickly. So I'll hopefully be able to talk to it a couple of times as it runs through. But, using automation, we can do a lot of control and precision manufacturing. We have polymer batch testing. We make our own polymer. We do the release, and we've seen that that's a highly repeatable process, even at scale. And we have a lot of tightly held tolerances that we can maintain with this automation, and include other algorithms to continually optimize the process. So I'll talk through it. So the robot's going over, taking it to another robot's measuring the base. So the white part is the former that we use. It's dipped in polymer there. And so that white piece is what drives the shape of the leaflet, takes it over. Another robot trims the free edge. So all three leaflets are formed at the same time. And here a robot separates the valve from the former and takes it and puts it into an inspection area where we can take photos and look for discontinuities or bubbles or things like that might happen from time to time, and make sure that we don't have any challenges there. So we can get a really consistent, good product each and every time through this. When we compare this to traditional manufacturing in this process, especially with bioprosthesis, there are a lot of steps that are involved in tissue valves. A lot of people need to be trained to make that happen, to go through all the process. There are several hundreds, if not thousands, of suture knots that are on each one of those valves. Take a long time to make those, test them, do packaging. We need to do packaging and sterilization as well, but a lot fewer stitches, and we can do that with robots as well with this. So manufacturing time is cut down significantly with something like this, and manufacturing costs. And so, this could potentially be a powerful tool to help, not just pediatric patients, but patients all over the globe, and looking at customized solutions, right? So we can get potentially in the future, it's not what we're doing now, but we could get a CT scan of a patient, go through and look at their, the size of their annulus. This one particular talk shows the aorta, and translate that into a design that's specific for that patient, and have the automation go through and make a valve specific for a patient. We don't see that as something that's too far off in the future. So we'll talk a little bit about, wrap this up and talk a little bit about our clinical validation. These results, we actually just had yesterday on the design part of this, yesterday a paper published in the Annals of Biomedical Engineering that talks about the design process of the Foldax valve. And a few weeks ago, we had another paper published in JACC on the clinical validation. So that's what I'm going to go over, just highlight some of this, but the full data is available in JACC, this 1-year study. So with our mitral valve, we did a trial in India, and 67 patients were enrolled in 8 cities. And that patient population ended up being 64% female. And 48% of those women were of childbearing age. Mean age is 42. So, I'll get to why that's relevant. But, we had excellent safety after a year, followed these patients for a year. There were no valve-related mortalities, no valve reinterventions, and no what's called SVDs, or structural valve deterioration. There are a number of reasons why these patients were implanted, but a lot of it had to do with rheumatic heart disease, which occurs in various areas of the world, specifically hits the mitral valve. And when a woman of childbearing age needs a heart valve replacement, typically at that age, they're given a mechanical valve. And with a mechanical valve, they need to be on anticoagulation for the rest of their life. What that means is that they typically can't conceive, or they shouldn't, because of the risks of being on anticoagulation. And there are things that they do to play around with anticoagulation that I won't get into. I'm not a clinician. However, what we've seen here is that, if we can reduce the dependence on anticoagulation, but still have a very long-term durable valve, that will help a lot of patients in these areas and that are affected by these diseases. So after 1 year, these patients, we monitor them, look at the pressure gradients across the valve and the effective orifice area. They go well. They're very stable. We look at the hemodynamics per size, and that's all very stable and looks great after a year. And we also look at the Kansas City score here, and their 6-minute walk test, and that improves over a year. That's great news. And their classification in their type of heart failure also improves over the course of a year. So that being said, the mitral valve was just approved for commercial use in India about 2 months ago. We started rolling that out commercially there. So that being said, thanks everybody for your time. That's what we have to talk about, and I think we'll have some time for some questions now.
Yes, we do. All right, very well done. Really intriguing presentation. And folks, I see some questions in the queue. Please send them along if you have them. I've got a few myself that we can sort of kick things off with, and then we can get into some of the ones in the box. But Ryan, you mentioned at some point the heart valve stayed open a little longer than the animal tissue valve. Is that what you said?
Yeah. Yep,
yeah.
Correct.
What's the significance of that? Is that better replicate the natural valve? Is there any significance to that, or is it just an observation?
Yeah, there is some significance to that, and what I did not show, but what we're working on gathering the data right now, is what that ends up doing is giving you more laminar flow, right? You get a big open orifice area, and what I showed over time, that the area of that valve, right, how it opens into this, I'd say, a quite circular cross section, means that you've got really good flow and really good flow over time that doesn't change over time. So that's the EOA, or the effective orifice area. But staying open longer, right, and having those leaflets, it's just less energy waste if you think about that, right? So more like the native tissue and the body, right? That you're not having to have a ventricle that's wasting energy trying to open and close. And I will say that, from an engineering standpoint, it isn't a huge amount of energy, right? We're not opening and closing a steel door or anything like that.
Oh, good to know, though. All right, a question here. This is probably for you, Jim, from Joe. What are the tensile properties, e.g., the tensile modulus for life polymer?
Yeah, as I responded to him already.
Okay.
I don't know off the top of my head, but
Okay.
the information is contained in that reference that I provided at the bottom of the slides on life polymer.
Okay, great. Sorry, I didn't know you had replied directly. All right. Another question. How many people are involved? Well, you mentioned the number of the two versus the 150. How the two people, are they merely observing like can you talk a little bit about the workflow with the robots, how they're involved, and what is a plant making valves for Foldax look like? Is it just a series of robots with a couple of people walking around? Describe the setting, please.
That's a great question. That's really what it is.
Wow.
People are surprised by, I would say, how small our footprint is. So the manufacturing cell that you saw there is basically 5 by 10 feet. And that's, what we call a single cell. There are cells outside of that in our clean room that are for preparation, right? Material flow and preparation. But ultimately, things go into this cell and the robots take over. They're monitored and, and watched, but we take data real time with
Hmm.
sensors as well. And so, the people that are there to monitor and watch and kind of do preparation and make sure material's flowing and everything's, going well. But the robots, they don't complain. They work nonstop if I want them to. Their fingers don't get tired, and they just go and go. And we give them a break at night when we have people go home. But it's a great little operation.
Great. And how do you see this playing out once you build plants and factories, assuming you go forward and you get all the approvals you need? Do you see the traditional setting where you have one large plant servicing a wide area, or do you see yourself setting up smaller plants regionally because you don't need to have a whole workforce? You just need to have a building with some robots in it.
Yeah, that's it could be done that way, obviously, and democratized and put anywhere. I mean, that's the beauty of automation, right? Doing automation projects on a variety of other applications in my career. You can put automation anywhere, and it's basically the cost of energy, right?
Mm-hmm.
And so you don't really need to worry about the cost of workforce so much. But they could be put anywhere, right? Locally. Our secret sauce and our core competence is actually in the polymer that Dr. Runt talked about. So that is, that's what we do in-house for sure.
Okay. So we have a question here from Mayara. She says, congratulations. Oh, they say congratulations on the excellent talk. I'm curious to know how the film processing works. Oops, sorry. Getting some questions and moved it along. I'm curious to know how the film processing works in the robotic fabrication process. Could you briefly explain how the system handles deposition, curing, and process control?
I'm reading the question while you're reading it too.
Number 4, yeah.
Yeah. So it's not really a film processing. We dip cast the material, and so the life polymer, it's a solvent based polymer, and so it's not like film processing. I don't know, Jim, if you want to say anything else on that.
Yeah, no, I think you handled that really well. It's not definitely not like film processing.
So, okay, it's about having a clean environment when it comes to, I think a couple of these things that were implied here, deposition, et cetera, anything else, process control, but that's all handled in part of, how the system is set up.
Okay. We have a question here. I'll ask for you, Jim, from Marco. Does Foldax synthesize life polymer in-house?
Yes, they do, in relatively large volume. And yes, it's polymerized with all the components in-house. I think now, Ryan, you guys are up to well over 200 large-scale batches, all of which have been characterized, and there's good repeatability between all of the batches in terms of molecular weight.
Yep, and there's anybody that knows polymers and does polymers knows that that's not an easy feat either.
No, it's not.
All right. Let's see. We've got a question from Farhad. Has any accelerated aging study been performed to simulate in vivo conditions, chemical stability of polymer?
Yeah, I would say there are accelerated aging. Well, let's see. So there's the accelerated durability that I talked about before, but I think that this applies more to the polymer. And I would again reference the article published by Chris Jenny, where a lot of that is discussed. But Jim, do you want to say anything else?
Yeah, just one first comment. There, one of the huge challenges in biomaterials or any materials environment is how do you know what's going to happen in 25 years without waiting for 25 years? And accelerated testing is fraught with problems. And accelerated testing is useful, obviously, but do we know for a given? This is a preamble. I'm not trying to dodge the question.
Filibuster.
I'll answer the question. Is raising the temperature by 10 degrees really associated with a time shift of, a couple of years or whatever the situation is? And you have to be super careful taking away the results of that and really predicting out in the future. Having said that, there are standardized, sort of standardized tests that are used to assess the hydrolytic stability and the oxidative stability of materials by really seriously for example, oxidation. There's a bench test that's supposed to correlate with a certain number of 6, 7, 8 years in service, which is a very aggressive oxidative, pretty high concentration of cobalt chloride and peroxide. And Life Polymer does extremely well under those conditions compared to other materials of like properties, of materials that are used in certain of this type of application. But yeah, it's tricky, right? It's really tricky. But yes, they've been done.
All right. Question from Cesar regarding in vitro durability, parentheses AWT, end parentheses. How much cycles the valve were submitted and how much to fail in DFM? I'm not sure if you guys can. Yep. Okay.
I can decipher that question. So that's exactly the slide that I presented before, right? So we take these in AWT, accelerated wear testing out to 600 million cycles, and that's what we take and submit for regulatory approval. That data, that's per ISO 5840. It actually goes above and beyond, but because we're polymer and a new material, we're special, so we get to do extra testing. Tissue valves and mechanical valves have different, I would say, a different requirement of 200 million or 400 million cycles, but we've done it up to 600 million cycles on this. And the second part of the question was on DFM, and so DFM testing is a little bit different than AWT. And so we take, you take that up to very high pressure for a shorter amount of cycles, not hundreds of millions, but tens of millions. And we do that testing as well.
Okay. We've got James asking, is testing of Foldax valves in pulse duplicators that open and close a valve per second worthwhile?
Well, that's what's prescribed by ISO and FDA. So yes, we do it.
I guess that's worthwhile.
We do it by both, both in real time and accelerated conditions.
From Justin, any plans to move this polymer onto an NITI frame for transfemoral delivery?
Yes, we are working on a transcatheter deployed valve right now.
Okay. And Marco asks, do you have any intention of commercializing Life Polymer as a standalone product?
I would say, the business is open to different applications. And so if there's something that you're interested or you think that it could work on, you're welcome to contact us.
Okay. And we'll do, this will be our final question, coming back to Mayara. How is anticoagulant therapy managed after the implantation of full access heart valves?
Great question. And so again, I'm not a clinician, but I'll tell you what I know from our clinical trial. So the patients, especially with mitral valve, if you know about this, you'll know that mitral valve patients have to be on anticoagulation for reasons other than their valve. They can have other issues that require them to be on anticoagulation, I'll just say. And so that being said, per standards that are out there from the American Heart Association, these patients were all on anticoagulation. The protocol was that they were prescribed anticoagulation for the first 6 months. And then it was up to the physicians to determine what the next steps would be.
All right. I think that covers everything. We're up against the clock. So Ryan and Jim, thank you for a really great presentation. Really, I think, get you thinking as to the possibilities for technologies like this, and actually very hopeful. So thanks so much for working on this.
Thanks very much.
Yeah, of course. Thank you for the opportunity. Really appreciate it, Tom.
Our pleasure. So folks, if you could push this along. There we go. If you take a moment, thank you. Yeah, please applause and thumbs up for our presenters. I think it was a fantastic talk. And once again, if you know someone who would have enjoyed this but wasn't able to watch live, they can still watch on demand. They can just go to devicetalks.com to register. Please take a moment to complete our survey. And I already said that. And all right, this is the series, Materials and Manufacturing. Thank you to our great sponsors, Bokers, Jabil, PSN Labs, Mitsubishi Chemical Group, Sekisui, and Forge Medical. And of course, thank you to you for being part of this Device Talk series. You can still register for tomorrow's live presentation by BioRetech. You should be able to find the link there on your page, or go to devicetalks.com. That'll be the final installment of this week's Materials and Manufacturing series. Thanks again for joining us and for being part of Device Talks.